Okay.
What number is this?
10. Okay, we're live.
We're good. Welcome everybody to World Harbor Project webinar number 10. So today we have another in the series of city-focused presentations, in this case from folks from Darwin in the north of Australia. And we're going to have two talks. The first is from Niels Munksgaard, who's going to talk about metals in Darwin Harbour. And then the second one will be about spatial and temporal patterns of microbial communities by Mirjam Kaestli. And Darwin is, I guess, a relatively new member of the World Harbor Project, but quite an interesting place. It's tropical in the way that a few of the other cities are, but it also kind of represents this vast expanse of the north of Australia. And there's a lot of talk in Australia these days about development of the north of Australia, and a lot of that potentially would be centered in or relate to Darwin. So an interesting place in the context of future plans for Australia. So with that, I'll turn it over to Niels, who will speak first, and followed by Mirjam, and then after that, as usual, we'll take questions. So over to you, Niels.
Thanks very much. Yes, and thanks to all the people who have tuned in. I'm going to talk about Darwin Harbour in the northern Australian region, and it's about metal and metalloid concentrations and how they have been influenced by urban development. And this is a collaboration with colleagues at the university and at the Northern Territory Department of the Environment and Natural Resources. So here we've got an image of the mouth of Darwin Harbour. We've got a large estuary down to the left-hand side and the open ocean out to the right-hand side. And you can see the city on a peninsula there. The motivation and aims of this study was to provide some reference data for development of sediment quality guidelines. So this study took place back in 2012, and another aim was to assess the spatial distribution of metals in both tidal flats, so that's in drying areas of the estuary, and also in mangrove creeks. As you can see in the image, mangrove creeks are a large part of the lining along the coast of the estuary. And lastly, we wanted to see if we could identify the sources of the metals. On the right-hand side there, you can see the estuary. So it's quite a large estuary, Darwin Harbour. We have Darwin City here and the large suburban area called Palmerston on the eastern side of the estuary. And the central and the western parts of the estuary are relatively undeveloped in comparison to the eastern side. So it's a drowned river valley, and we've got macrotides up to 8 metres at spring tides. The catchment itself is quite small. That's the light grey area here. And the small size of the catchment compared to the estuary itself means that there's a reduced potential for disturbance. In total, the urban, industrial and agricultural areas comprise about 13% only of the catchment, but currently there's some major expansion happening in the oil and gas industry. Sediment fluxes are mainly directed up estuary by the tides, and the net export to the ocean is quite small. We have a monsoonal climate, so there's like a dry season and a wet season. Almost 90% of the rainfall falls between December and March or maybe April. In the upper arms of the estuary is where most of the environmental concern might be. because they're fairly poorly flushed. So whatever runs out tends to come back up again. As I mentioned, this was done in 2012, and it was sediment sampling on intertidal flats and mangrove-lined creeks. Here's a map of the sampling points, about 250 sites, about 300 samples, and a number of sites were done in triplicate to gauge the variation in concentrations within each site. You see here the division into the eastern side, the middle side, and the western side that we're going to come back to when we discuss the results. You can see in dark green all the mangrove areas. You can see the urban areas, and these are the dry land areas around. The sampling was done with using a pole coring device that could reach up to 3.5 meters of water depth, and we focused all the sampling on the top 0 to 5 centimeter sediment depth, so all was sampled consistently. We analysed grain size distribution by laser diffraction. We sieved the samples to a less than 2 millimeter fraction, which is basically almost always all the sediment sample. And we digested the sediments in 2 strong acids at high temperature, so we get what we call a near total concentration. It's a total concentration for these types of sediments that are mainly dominated by clay minerals and carbonates. Metal analysis was carried out by ISPMS, and we did a couple of other nitrogen, sulfur, and carbon analysis as well. Grain size distribution influences metal content. This is because very fine-grained sediments, clays and silts have a large surface area, and there's a lot of absorption sites for the metals. And therefore, if you've got a situation with greatly varying grain size across the whole of a large estuary, you need to normalize for the influence of the grain size on the metal content if you want to look at the distribution and tracking of sources. So we could show that the aluminium content and the clay and silt fraction were very strongly correlated, as seen here in the left-hand figure. So up here we have clay-rich compositions which have a high aluminium concentration of some 8 to 10%, so 80,000 to 100,000 milligrams per kilo, with the equivalent of 8 to 10 weight percent. And there's a very strong correlation here with the grain size distribution. There's another normalization method called the pen method, but that was unsuitable in this case, and that's because low aluminium occurs for 2 reasons. It could either be because there's more quartz in the sediment, or it could be because there's more carbonate. So there's not a straightforward correlation. If we plotted our compositional data in a diagram that's designed to tell us about the normative mineralogy, we can deduce that our sediments are mainly composed of clays, kaolinite and smectite. The second major group is the iron oxyhydroxides, and the third major group is calcium carbonate, calcite and aragonite. And we can see that across our estuary, these are all the different points here, we have a huge variety of normal or normative mineralogies, depending on where we are, depending on the energy levels of the estuary, etc. So this also shows you the importance of normalizing for this variation in terms of grain size distribution. So we use aluminium to normalize our grain size effect. So we use it as a proxy for the fine grain sizes. And here we have 2 sets of diagrams. On the left-hand side, we got copper, and copper divided by aluminium concentration, and we've got zinc, and zinc divided by aluminium concentration. And we can see that when we do the normalization, we reduce the spread of data. So this is concentrations along the x-axis. We reduce this spread and the bias or the skewness in the data when we do the normalization. So normalization is at the bottom here. The same with zinc. We've got a skewed diagram of frequency when we use the direct concentration of zinc. When we normalize for aluminium, we reduce that skewness. We then produce the number of maps showing the distribution of our analytes, and we can see here that on the left, the clay and silt map looks very similar to the aluminium map. And this is, of course, because they are very well correlated, as I showed before. We then had as major products of our study a production of a whole range of metal concentration maps, where the size of the circle reflected the concentration of the metal of interest. We have here a map of straight copper concentration on the left-hand side, and we can see that there's like a pretty noisy distribution around most of the harbour. It's not a pattern that's all that clear. When we normalise for grain size via our use of the aluminium proxy, we see a much clearer picture. We see a stronger signal, larger circles of copper at the developed eastern side of Darwin Harbour compared to the middle and western side, which have basically the background level of aluminium, of copper. This shows you the value of normalization. We should mention that none of the sites, and this is looking at the raw concentration, none of the sites exceeded the so-called ANZAC ISQG low levels. These are the National Australian Sediment Quality Guideline levels, which are set at 65 milligrams per kilo. Our maximum is 23. So although we can see the influence of the city, we're still pretty much at low levels. We're at near pristine levels. Similarly for zinc maps, on the left the raw zinc concentration, on the right the normalized zinc concentration. Again, we can see a clear pattern of increased concentrations on the right-hand side when we normalize for grain size. And again, we just sneak under the ANZAC guideline of 200 milligrams per kilo zinc. Our maximum is 190, but the mean is much lower. So we have also a pretty low, presumably non-toxic level of zinc in the estuary. The ANZAC ISQG low level is set such that you would expect toxicity to not be a problem when you're below that level. Here we've got 6 metals compared between the city area to the east, the middle arm, which is the middle of the estuary, and the western side. We can see zinc here in a box and whisker plot is enhanced, increased on the city side. The same with lead, cadmium, not so much cobalt, not so much nickel, and a little bit with chromium. So for those who might wonder, the box and whiskers plot, the middle line is the median, the box is the interquartile range, so 25 to 50% percentiles, and the bottom and top are the lower and upper ranges. We ran some statistical software over this data, and using a method called the generalized estimating equation modelling for the sediment, we arrived at these sort of conclusions here, and these are all statistically significant findings. So when we compared copper in the city and with other areas, so non-city areas, we had a 28% increase in the city. We had a 20% increase in lead in the city. We had a 29% increase in zinc in the city. And when we go to compare intertidal with mangrove areas, we had 42% less copper in the intertidal areas, 39% less copper lead in the intertidal area, and 44% less zinc in the intertidal area, all compared to the mangrove areas. And out here to the right, we see the level of metal that increases with each weight percent of aluminium. So this again reflects the influence of aluminium-rich, in other words, fine-grained sediment. So they all increase, all these metals here, significantly, except for zinc down here, which was non-significant. So the second part of my talk is something we did a couple of years later in 2014. We wanted to investigate a small subcatchment and try to find out the history of when this metal increase from urban areas occurred. So we looked at a subcatchment called Rapid Creek, and this is where the Darwin Airport was developed during the Second World War. So here's the airport, and there's northern suburbs, which a lot of these were developed much later than the Second World War. And we called down here in the estuarine section where there's a tidal influence, and here on the right-hand side is a blow-up, and we took 4 cores in this area, 4 sediment cores, down where the freshwater flow slows down and where sediment will settle under low energy conditions. So we took 4 sediment cores that were down to 80 centimetre depth, and they were sliced at 20 millimetre intervals to carry out metal and lead isotope analysis. And from one of the cores we did 10 millimetre slices in the top 23 centimetres, and they were sent off to ANSTO at Sydney for the dating method that's called the lead 210 method. And they carried out some optical and X-ray scans, which is what we see here on the right-hand side. So the optical image is in the background of the core, and the X-ray scan is here on the right-hand, in the centre. And this is the 23 centimetre depth level. We were advised that only one core was suitable for age dating, so it's an expensive exercise, and we only did that one core for that reason. But the other 4 cores, we did a range of metal concentration analysis, and if we focus here on the blue line, that's the core that was sent away for dating. So this is the one called RC1, and we can see here for copper over aluminium that at 15 centimetre depth there was a sharp increase in the concentration. It basically doubled compared to what was lower and therefore older. The same for zinc. At 15 centimetre we see a sharp increase, cadmium increased, and lead increased as well. The other cores here, we can see that the green and the red one, they must be fairly young, all the depth of those cores, because they have the elevated level for most of the metals. And we can see that the purple here is not really clear how that compares to the other 3, but that was up on a floodplain which has some desiccation cracking, and that was probably some mixing occurring there. We also did lead isotope ratios. For those who are not familiar with those, and these are different from the lead-210 dating method, but lead-28 and lead-27 was of relatively high abundance in lead that was used in Australian fuel for many decades, and therefore runoff bringing in lead from the catchment during that time period will have increased ratios of 208 to 206 lead and 207 to 206 lead. And you can see here for the blue core again, at 15 centimetres there was a sharp increase in the ratio, signifying the start of lead coming into the core area this is lead that was influenced by the addition of leaded fuel into the runoff. And the other two cores here again show a high level all through the depths, except maybe the very low end, deep end of the green one. Finally, we got the age dating, and there's two ways of evaluating your 210 lead dating. The two models that you can choose between. Now, we naturally chose the CRS model because it fitted what we knew about the development time of Darwin Airport. And at 15 centimetres, which was where we saw the increased metal levels and in lead isotope ratios, we found an age of 77 plus or minus 9 years, which corresponds very well to the known development of Darwin Airport just prior to and at the beginning of the Second World War. And that should pretty much conclude my talk here. So in Darwin Harbour, intertidal sediment, we found that the main metal-bearing minerals are clays and iron oxyhydroxides. There's strong positive correlations between clay and silt, grain sizes and metals. We used aluminium normalization to correct for the confounding effect of grain size, and that allows us to seek out the sources, define the sources and the distribution of metals in the estuary, and we showed that there was increased levels on the developed eastern side of Darwin Harbour at a size of 20 to 29% for these three metals, copper, zinc and lead. But we also noticed that there is virtually no exceedances of the NSEC low level guidelines, which signify the start of toxicity effects. Therefore, our final conclusion is that still large parts of the Darwin Harbour estuary remains in a near pristine condition with respect to metals. So thanks very much for listening to that, and that concludes my talk.
Great, thanks, Niels. So when people talk about measuring contaminants in some very broad sense in urban waterways, you know, they usually talk about two things. They talk about chemicals, including metals, as we just heard from Niels, and then the other thing that they talk about increasingly are microbial communities, particularly given the increase in our capacity to describe microbial communities. So we've heard about metals, the chemical part of this, from Niels, and so now I'd like to introduce Mirjam Kaestli again to talk on the microbial part of it. Over to you, Mirjam.
Okay, thank you very much. So yes, we've already heard from Niels about, just getting this going, here we go, about Darwin Harbour and where it is. So we'll go through this initial part quite quickly. What is important for the microbiota patterns is it is in the wet-dry tropics, so we will probably have some strong seasonal patterns, and it is mangrove lined, and it is a macrotidal estuary. So as we heard, there's a large tidal range up to 7.5, 8 meters, which means that there is some really strong tidal forces, particularly during the spring tides. We also experience, or Darwin Harbour experiences, increasing urbanisation. There are about 130,000 people living in its watershed, and secondary treated effluent is the main point source of nutrients, while diffuse urban runoff probably makes about 20% of the anthropogenic nitrogen input. And so, of course, with this increasing urbanization, there is a need to monitor the ecosystem or the harbor health, and microbes or bacteria are a good target, being abundant, easy to measure, high diversity, and also being responsible for quite a few biogeochemical processes. But the problem is that there's actually not much known about the microbiota response to nutrient load in tropical estuaries with the strong hydrodynamic forces such as the seasonal forces and the tidal forces. So the study, our study objectives were first the analysis of the spatial and temporal patterns of the microbiota in water and sediment of Darwin Harbour. Then we intended to examine the association of the bacteria with abiotic factors. These are mainly nutrients. Then determine whether there is a human impact signal there at all in the microbial patterns despite of these strong mixing forces. And ultimately, the aim of our project is to find microbial taxa and functional markers which are an indicator for effluent in this ecosystem. So our study approach was a single impact double control design. So we had all up 32 sites across 2 impacted and 4 reference creeks. And so the 2 impacted creeks were Buffalo Creek in the north in Charles Bay and Myrmidon Creek in the south of the harbour in East Arm. And for each impacted creek, we also had the 2 reference creeks. So I should probably say that with impacted creek, this means these 2 creeks received secondary treated effluent from 2 or each one wastewater stabilization pond, and they also received or receive urban runoff, in particular Buffalo Creek. So we did the sampling over 8 rounds. This means over 2 years in 2 dry and wet seasons. We always sampled at high tide during spring and neap tides for each round. And all up, we collected 286 water and 208 sediment samples. So we measured the microbiota, like the bacteria, through 16S RNA gene sequencing, and we measured various water and sediment FISCAM factors as well as nutrients. So what did we find? As to no surprise, we found that the temporal factors, which are seasons, years, tides, were indeed really strong, and they had a stronger impact on the water, especially the water, but also the sediment microbiota than spatial factors. And in particular, seasons had a very strong impact for the water microbiota. And you can see this also on this PCO ordination on the right-hand side. So for those of you who are not familiar with this type of ordination, every dot signifies a bacterial community or a sample or a site, and the closer the dots, the more similar or the more related are the microbial communities. So this PCO shows all the water bacteria from East Arm, from the southern part of Darwin Harbour, and you can see a very clear clustering according to rounds of samples, in particular according to seasons. We have the dry season, the orange and brown samples at the top, and we have the blue, the wet season samples at the bottom. But very much to our delight, there were not just temporal factors, but the spatial factors also were we saw quite a clear signal there. And what you can see here is the average bacterial community per site for the neap water. And you can see 2 trajectories, brown and the green one, and they are for both impacted creeks. So the brown one here is for Buffalo Creek, and the green one here is for Myrmidon Creek. And they always start at the most upstream sample and go down to the mouth of the creek, with the reference sites here. So what can we see here? So for instance, we see that the bacterial community at the urban runoff was quite distinct, and it was also quite distinct and different from the treated effluent. And both treated effluents from both water treatment ponds, like the one close to Buffalo Creek and the one close to Myrmidon Creek, they weren't exactly the same, so the microbiota differed between those. And now what is quite interesting for Buffalo Creek is that Buffalo Creek is quite a narrow, meandering channel, and it has a large sandbank at its mouth, which leads to very slow water mixing. And this is really nicely reflected in the bacteria. So you can see the sites here, or the microbial communities here, which really only slowly revert back to our background community we have with the reference sites. For Myrmidon Creek, this situation is quite different. So the outfall, or the effluent, treated effluent, enters the creek further down towards its mouth, and just in general the channel is much wider and there is good mixing. And we really only have a clear human impact signal at this site 3B here, which is a small side channel where the effluent enters the creek. So in a way we have a typical disturbance recovery signal for the Myrmidon Creek. What is also interesting is that for the reference sites, we had the distance from the most upstream site to the mouth was actually quite similar for all, or the same for all creeks, including reference and impact creeks. But we really only saw this differential signal, spatial signal for the impacted creeks, whereas the reference sites, they all had quite a similar microbial community, no matter where in the creek. So I mentioned before that this is the neap type water which I showed you, but so why didn't I show you the whole water microbiota? Well, the fact is that, as mentioned, there were strong temporal factors, and one of them was tides. And the tides indeed had a strong impact on the spatial signature. So while the neap tide, we saw some really nice spatial signals, we did not see this during the spring tide, where we really, the only thing we saw was a clear urban runoff signal again. We saw the outfall signal again, but all the other sites clustered quite tightly, which is not surprising given the really strong mixing we have with the macrotidal spring tides. So on this slide here, it's a summary, similar summary to what I showed you before, again on the average spatial signal of the microbiota. But this time this is shown in form of a phylogenetic tree. And we can also see the pie charts with our main average bacterial phyla per area. So there's several things we can see here. One is we again have this we have this very distinct branch in the middle of the tree, which shows our outfalls or treated effluents, again, which slightly differ between the two treatment ponds. And they are, again, most similar to our urban runoff here. But what we also can see is that they, again, are most similar to our impacted creek, Buffalo Creek, with the slow mixing, which makes sense. And I should also mention that when we have a look at the pie charts, that the effluent impacted sites, they had a larger proportion of cyanobacteria, which is not surprising given the amount of nutrients. And they also had our green sulfur bacteria, the chlorobi. And we can even see a small red slice here. These are the firmicutes, which are well known, or which have taxa, which are well known inhabitants of mammalian guts. Finally, what is also interesting here is that the southern part here, east arm, there's hardly any spatial structure what we've seen before, and east arm is as I mentioned before, has much better mixing, and it's just still more pristine than Buffalo Creek. And the wastewater treatment pond has not been there for as long as it has at Buffalo Creek. So again, we see that there is much less of an impact here. So now we've seen the spatial and the temporal signals, but how does this relate to the nutrients? Here you again see an ordination with the site average bacterial communities, but now you also see the correlation factors for nitrates, ammonia, and salinity, and you see the non-linear or the contour lines of these nutrients and salinity, which were fitted with a non-linear GAM model. And there's several things you can see here. One is that the urban runoff sites, so the bacterial community there was associated with the highest nitrate levels, whereas at outfall, these communities were associated with the highest ammonia levels. And then, similar to before, we have our impacted creek microbes or bacteria, which only slowly revert to our background signatures along the nitrate and in particular along the ammonia gradient, back to our background signature, background sites. So far we know that, yes, there is an impact, a human impact signal. We know, yes, the bacteria are associated with nutrients. Yes, they are associated with salinity. But we don't know, coming back to the human impact signal, whether that's mainly due to nutrients or due to freshwater inputs, in other words, changes in salinity. So that's why we did a variation partitioning analysis where we distinguished between salinity, nutrients, and spatial eigenvector impacts or associations. And we saw that indeed the nutrients explained the most of the explainable variation in the microbes for both East Armandjoal Bay, followed by salinity. So far, I've only talked about the water microbiota, so you might wonder, what about the sediment microbiota? And this is, again, a side-averaged ordination of the sediment microbiota, where we have, again, our trajectories for our highly impacted, the Buffalo Creek, and the less impacted, the Myrmidon Creek. And as you can see, there is no clear spatial signal here. So the only communities which clearly stand out are our our effluent associated outfall communities, as well as these upper Buffalo Creek communities. So they're mainly those communities with a history also of high impact. As I mentioned before, Buffalo Creek had its water treatment plant for longer, for 4 decades, and so it seems we might actually also see this here. And we have hardly no creek-specific signature for the more pristine east arm, which are the circles here. We also found that there was no strong association with the nutrients we measured on the day. And it is known, and it seems our data supports this, that the sediment microbiota reflects more current and past conditions, whereas for the water we really get this current snapshot at the time. So we now come to the last objective, which was using these temporal and spatial patterns we found and see whether, based on this data, we now can develop some microbial bioindicators for human effluent, which are suitable for Darwin Harbour. And you might wonder, why can't we just use some well-established markers such as enterococci? Well, enterococci commonly occur in urban runoff in Darwin, and it is known that they persist in nutrient-rich warm waters in the tropics. And this is just an example based on our 16S data. So these are the enterococcaceae, and you can see it here that we had actually lots of enterococcaceae in the urban runoff, but hardly any in the human effluent. Whereas if we compare this to, say, for instance, ruminococcaceae, which are also firmicutes and which are known to also occur in the mammalian guts, we have a much better association with actual effluent, where we had the highest load in the treated effluent and much less in the urban runoff. So one way to track effluent could be to not just focus on one bacterial taxa, but actually focus on the whole microbiota by using Bayesian source tracking. And so here's an example. We used urban runoff, or say land runoff, as one of the sources, and treated effluent or outfall as another source. And what we can see here, so this is from the dry season in 2013, we can see that as we go down towards the mouth, the contributions of the outfall get smaller and smaller. And it is overall similar between the neap and the spring tides. Maybe it is a bit bigger in the neap tides, but also we see a relatively large contribution of land runoff as compared to the treated effluent. Now, we've done the same exercise during the wet season, and it does look quite different. So the green part, the outfall or treated effluent contributions, are considerably larger for Buffalo Creek, in particular during the neap tides. And considering that this is a wastewater stabilization pond which is exposed to the monsoonal rainfall, this might not be surprising. So another way, and this is the last part of how we approached the bioindicators, is to also look at the functional bioindicators, to look at human impact, in particular at the nutrient load, like nutrient inputs. So in tropical estuaries, the impact of nutrient load on ecosystem function, such as the nitrogen cycle, is still unclear. And you can see here the nitrogen cycle on the right-hand side. It is mainly driven by microbes, and you can see the some of the genes which are mainly responsible for the enzymes which drive this cycle. So we collaborated with Lev Bodros's group at CSIRO Hobart to use their novel developed nitrogen cycle gene microarray to compare the nitrogen cycle genes between the sites with human impact and our reference sites. And so what you can see here is the results of this microarray. So this is a heat map. And I should also mention this is now the sediment. So one thing we saw is that there were very few signals in the water, and it is known that the nitrogen cycle is mainly occurs in the sediment. So this is all the sediment samples. And so all the different columns are the different nitrogen cycle gene probes. So for instance, they might be on AMO-A, like nitrification genes or enzymes, or they might be on NIF-H, which codes for nitrogen fixing. And all the different rows indicate the different samples, where we start with our high impact outfall related samples and have our reference samples down here. And so for instance, what you can see here is that we had far more signal from archaeal ammonia oxidizers than we had from bacterial ammonia oxidizers. You can also get a bit of a hint that there is less archaeal ammonia oxidizers in the highly impacted as there was in the reference sites. So the analysis of this data is still very much ongoing, but one of the aims was to now find those probes which showed the clearest difference between highly impacted and control sites. And I show you two examples. One of the examples was the archaeal MOA nitrification gene, which codes for ammonia monooxygenase and codes for an enzyme, or that enzyme which does the nitrification from ammonia and oxidizes ammonia to nitrites. And the probe we found which really differs between highly impacted and reference sites belongs to the archaeal nitrosopumilus. And we designed the qPCR assay targeting that probe, and we found indeed that this is now the results of the qPCR assay on the sediment samples, that there were more nitrosopumilus AMO-A genes in the control sediment as they were in the highly impacted sediment. And this is not surprising, because nitrosopumilus is known to be adapted to highly nutrient limiting conditions. And yes, so further validation is ongoing on this. So we also looked at NOSET, which encodes the nitrous oxide reductase, which is the last step in the denitrification, which takes nitrates out of the system and turns it into nitrogen gas. So we again had designed a qPCR for NOSET genes, and we found that it was highly reduced at highly impacted sites, and this was despite an abundance of inorganic nitrogen. Now, this sediment is now not the sediment we used or from our study, but this is actually independent new sediment from the same sites, though. But this sediment are samples from Julia Fortune's PhD project. So Julia Fortune is a PhD candidate and senior researcher at the NT government, and she did some extensive sediment flux studies with these samples, and she found a reduced denitrification efficiency in the samples, or at the sites we found less NOSET genes. And it is a previous study on these sites has also found that there is a reduced or highly variable denitrification efficiency at these sites, and that study was by Jody Smith. And it is hypothesized or assumed that this might be due to a breakdown in the coupling of denitrification and nitrification, because these are highly hypoxic sites and there's also sulfides present. And it could also be that other processes compete for the nitrate, such as DNRA and like dissimilatory ammonification, or yes, and also benthic algae, which might also compete for the nitrates. So we found indeed not only that it was reduced DE and NOSET at the highly impacted sites, but there was overall a weak correlation between these two processes. And we would not expect a strong correlation, because it is known that the denitrification efficiency is very much dependent on the nutrients availability on the day, and so it would be highly variable from between different time points. And this is also what Julia Fortune saw, a high temporal variability, whereas the NOSET gene, or the presence of the bacteria in the sediment, that's more reflecting an overall condition, like it goes basically back to what we discussed prior, that the sediment microbiota more reflects present and past conditions. So we were happy about these results, but there's still more work to do. So our outlook is that we want to refine the nitrogen cycle qPCR assays further. We want to validate the assays with independent new sediment samples, validate the assays not only for gene abundance, but also for gene expression, like look at the mRNA or the cDNA levels, and validate the assays further with the sediment flux data by Julia Fortune. So our take-home messages is that the seasons and the tides had the strongest relationship to the water bacteria, which nicely reflects its location, or the location of Darwin Harbour in the wet-dry tropics, and the macrotidal nature of the estuary. Then we found that the knee border microbiota showed the clearest spatial pattern, whereas the sediment microbiota seemed to reflect past and current conditions. And the differences we saw in the bacterial patterns between Darwin Harbour, they seem to reflect the harbour's complex hydrodynamics and bathymetry. And we were happy to see we did see a clear human impact signature or signal in the microbiota, so it was limited, and overall the Darwin Harbour still seems to be in a good health. And we have some candidates for bio or functional bioindicators, like the nitrosopumilus AMO-A gene, or the NOSET gene abundance, which we found to be associated or correlated with the sediment flux denitrification efficiency. And these are all the people I would like to thank, and who were very much worked hard on this project. Thank you very much.
Fantastic. Thank you, Mirjam. We've heard in detail about both the chemical and microbial side of things in Darwin Harbour. So any questions for Mirjam or Niels? You'll recall so are you about to turn them on?
Is there a question?
So questions for either Mirjam or Niels from the audience? Well, while people across the world are pondering, I'll go ahead and ask one. So I guess it seems like an obvious question to ask for the sediment microbiota. If the two of you have combined forces and tried to figure out whether there's any influence of metal composition in the sediments on the microbiota or any relationship.
We also collected metal data, and we did not find that strong associations. We found some association with copper, and that was mainly at the highly impacted sites, and also with sulfur, that was mainly at Buffalo Creek, again, which was more impacted.
Right. I mean, so do you think, I mean, overall your metal concentrations, as Niels was saying, were pretty low. So do you think that's the main reason that you're just not getting much of a relationship?
Yeah, I think this makes very much sense. And it also, again, goes back to that, especially East Arm is still in quite a pristine conditions, and we found overall quite low levels there at the sites which we collected the samples from.
The other quick question I had also about the microbiology was that people are starting to use the integrase genes as a signature of human impact, urban impact. Have you thought about using those markers in Darwin?
Not yet, but I definitely, yes, sounds like a good point.
Cool. Anybody else chiming in? Looks like we got a few people who are still yeah, we're just
oh, there we go. I'll put her on.
Okay. Karen, I think you're about to go on.
We can hear you now, Karen.
Karen?
As in Karen Gibb?
Yeah, that'd be the one.
Okay. So this is actually a question for Niels.
Yeah, yep, I'm here.
It relates to Peter's query about the metals and the microbes. So do you think it would be more interesting, or you might tease out patterns if you separated total from bioavailable metals in sediment?
The relevant concentration is, of course, the bioavailable ones, but we should keep in mind that if the total concentrations were relatively low, but well below expected toxicity levels, we would expect the bioavailable portion to be even lower. So at the moment, we probably would have difficulty in showing any correlations with the microbiota, maybe except for some very small localized area that we could seek out. But sort of harbour-wide, I think there'd be very little hope of seeing any correlation so far.
Right. Fair enough, thanks.
Anybody else? I guess one more for me while we've got you. So one of the things that we've started talking about in the World Harbour Project is, as Karen knows, and I guess as the two of you knows, is a microbial project, and that's starting to take some shape, and I think will be discussed further in Xiamen. And in particular, we're interested in comparing traditional measures of microbial contamination in waterways with some of these, some of the more modern and more accurate sequencing or qPCR methods. Do you by any chance have coliform counts or equivalent with some of the microbial sampling that you did, or was it all sequencing in qPCR?
For our study, it was all sequencing, but the NT government is doing routine routine coliform counts in many sites throughout the harbour. So there's definitely data around we could compare it to.
Right, and that's ongoing. To
my knowledge, it is. Karen and Niels, are you still? If you're still.
Yeah, I'm still here. I don't really know exactly what the NT government is doing, but I would think that they have obligations, at least in some parts, to continue microbial sampling. I know that they do sediment sampling for metals, maybe annual interval. Some of Buffalo Creek and possibly Myrmidon Creek. Yeah.
So it's on my list of things to do to get back to, in particular, Karen and Mirjam about some of the details of this. It probably now won't happen until after Xiaomen, but you know, maybe to just if you have the time to explore a little bit about whether they are taking standard water samples for coliform counts, and whether it would be possible to piggyback some water sampling on the back of that, which we could then take and do some more sophisticated things with. You know, maybe if you wanted to talk to your contacts at, is it NT Water? Is that the relevant agency? Then that could be helpful in terms of us thinking about what this project might look like.
Yeah, we can definitely look into that. It would be the aquatic health unit at the NT government.
NT Aquatic Health Unit, okay. Yeah, whatever the relevant agency is.
Yeah.
Excellent. I do see a list of people who are listening in, so any further questions? Speak now, you know, forever hold, all that sort of stuff.
Peter, I have a question. I'm at Bill Dennison. I'm in Karen Gibbs' office right now. And
are you?
Yeah, and so I down the hall from these guys. But one of the things that we were talking about this morning is the rapid rate of relative sea level rise in Darwin Harbour. It's really prodigious sea level rise rates. And I was wondering if the runoff patterns that you're detecting with both the microbes and the metals, whether you would anticipate a change in those patterns as you start to inundate the mangroves and the outfall and runoff patterns from land use would be impacted by that sea level rise.
Yes, I could definitely see that there would be changes. And when it comes to the land runoff, there have also been some more recent modeling done, which suggests that there is actually much higher contributions from the land runoff, say also nutrient contributions than was initially or was modeled some years ago. So this is definitely on the radar, and I don't think this did take into consideration the sea level rise, so it probably would then be even higher. But yeah, and also with the mangroves obviously then being more inundated and changing biogeochemical processes there too. Yeah.
I was thinking maybe because you mentioned sulfur perhaps affecting the denitrification coupling, and that sea level could bring more sulfate into those areas and sort of extend that lack of denitrification impact inland.
Yeah, this makes sense. Yeah, in which case denitrification would be disturbed or would be inhibited even more, or the nitrification coupling. Yeah.
Are there any predictions about whether the amplitude of the tides are predicted to change under future climate scenarios for Darwin Harbour? Do you know that? I
don't.
No.
I've never heard of that anywhere, Peter. In terms of you know, that certainly the average, you know, sea level rise is the average, but whether the extremes would be, you know, maybe more stronger springs or
Yeah, no, it just occurred to me I had never heard anything about that either. I mean, in light of all the things that are happening in the U.S. at the moment, you know, I guess if some of the stronger tides coincided with some of these extreme events, you know, they could act in concert with each other. But just in terms of affecting tidal cycles, I don't know that I've seen anybody do that.
Well, the tides are mainly, you know, driven by astronomical things and modified by the positioning of coastlines and continents and so on.
Yeah, all that stuff. It's true. I mean, but others. I mean,
why arise should change the tidal cycle.
I was thinking more about the amplitude as opposed to the cycle, but anyway, I'll take that on notice.
Do we have Jeffrey?
I don't know. Is that a question from Jeffrey?
Jeffrey, or do you have a question?
No? Okay. Well, I think we've reached the end of the hour. So, Mirjam, Niels, thanks very much. Great stuff, and yeah, look forward to pursuing in particular some of those microbial pieces, Mirjam, as we were talking about. And Niels, as you probably know, there's been a bunch of processing of some of the contaminant data from a bunch of the World Harbor Project harbors back here through these particular risk models. So one of the things that Edwina and I were talking about offline was just the way that you process your data versus the way that we've processed it here. So I think that's a conversation that we'll probably want to get back to you on. So you'll be hearing from either John or Edwina. So definitely things to follow up on. But thanks to both of you. Thanks to all the folks who beamed in. So the next webinar would nominally be for the last week in September, but there's not one scheduled because all of us will be up in Xiamen for the project partners meeting. So the next webinar is scheduled for after the meeting, I'm told, and so we'll get back to you on specific. So thanks again, good to talk, and we'll talk again either in Xiamen or at the next webinar.
Yeah, thanks for the opportunity.
Thank you. Thank you.
Hey, folks.
Okay,